Hi everyone, I am Manling and my project is about measuring the enzyme activity of various mutant strains of galactose oxidase. These enzymes speed up the oxidation of alcohols into ketones, and hence by measuring ketone yield, enzyme activity can also be determined. Those oxidase reactions usually take place in water. High-performance liquid chromatography is favored for ketone yield quantification. However, for non-UV reactive compounds like 3-hydroxy-tetrahydrofuran, this method cannot be used, and hence gas chromatography mass spectrometry or GCMS will be used instead. My experiment consists of parameters optimization, liquid-to-liquid extraction, as well as the construction of a calibration curve. The parameters optimization phase, temperature, flow rate, injection volume, and split ratio had to be determined. The goal was to separate ketone peak from other peaks like the alcohol, solvent, and dimethyl sulfoxide, which is found in the reaction mixture, with adequate sensitivity for quantification. Abundance was then quantified by taking the area under the curve using computer integration, and these are the optimized parameters. Next, liquid-to-liquid extraction is necessary because the reaction mixture consists of water, and this cannot be directly injected into the GCMS as this will result in poor peak shapes, which makes it very difficult to quantify ketone yield. Ethyl acetate demonstrated highest extraction and hence was chosen as the solvent. A calibration curve was then made for the area of ketone for various concentrations, and the final results were as hypothesized. So this optimized GCMS method can be used for structurally similar ketones. Additionally, results can also be compared with a wider range of substrates, so machine learning can be employed to develop an integrated enzyme model to reduce enzyme engineering speed in industries.